Okay, and let's talk proteasome. The proteasome is like a giant protease that's really good at cutting proteins in lots of different ways. The proteasome is your personal protein shredding device. It's a large, multi subunit protein. And the multi subunit part is important because we can change out some of the subunits when the cell is actually under attack. And the regular proteasome will chop proteins at certain places. And so it can only present a certain set of epitopes. But when there's actual danger, you can make a different version of the proteasome called the immunoproteasome, and this thing will actually cut proteins in different ways. And so potentially it can give you another avenue to present little peptides to your immune system. So let's go. Let's try it. All right. So the two parts of the proteasome are the 20S catalytic core and the 19S regulatory cap. And the cap is just like it says. This is a bit that locks onto the ends. The whole protein is kind of like a cylinder. And the 20S catalytic core, this is the bit that actually shreds proteins that go inside. So it has a chamber in the middle that kind of looks like an hourglass. I think we got some good pictures of that coming up. And yeah, it unfolds protein, it chops it into little tiny bits, and then it opens the caps and spits out the little bits. Okay, so the subunits, there's some subunits in the beta section. So these are in the catalytic chamber, and you can change out beta 1, 2, and 5. And if you change out those to different versions of beta 1, 2, and 5, then you can make different specificity. The proteasome will cut something different than it would have cut originally. And so I think we'll get to those on the next slide, but I think they're beta 1 star, 2 star, and 5 star. But let's see. Let's see what they are. Okay, and then you've also got the ubiquitin proteasome system. And so if you put a ubiquitin linked by its lysine number 48 on the ubiquitin, because there's a couple places you can attach this to a protein, that is a quality control system. So proteins that are made but don't fold up very quickly in the ER, you'll try and glycosylate them, deglycosylate them in the Golgi or whatever, but if they just do not make the grade, then you're going to stick a K48 linked ubiquitin on them, and that just is like, I'm sorry, that's the failing grade. That's your cell can give to a protein, basically. It just says that this one has got out of hand. Something's wrong here. Let's degrade this and start over. And there's always another chance in the cells. So that's good. Okay. Oh, I was so close. Yeah. So beta 1, 2, and 5, I are the immunoproteasome versions of that. I said star, but it's not. It's I. And do they have other names? Of course they do. This is cell biology. Everything's got a couple names inside the cell. Do we need to learn the other names? Nah, we don't need to learn those. Just know that beta 1, 2, and 5 are the subunits that you can change out on a proteasome. So ubiquitination is adding a thing called ubiquitin to a protein. And then once the protein is tagged with ubiquitin, then it's going to have a different function. So, for example, we had those polyubiquitin chains. You remember those? In some of the TLR activation pathways. And ubiquitin can also be used to mark proteins for recycling. It can it's just like a glue that can hold different proteins together. Sometimes it's necessary for inflammatory response. Yeah, ubiquitin does a lot. There are a couple of other proteins that work almost exactly like ubiquitin. So one of these is called SUMO, SUMO. And so adding SUMO to things, it looks just like ubiquitin, honestly. It's called SUMOylation, and it's going to have its own set of effects, which are going to be overlapping with ubiquitination. And there's also another thing called ISG15, which is an interferon stimulated gene number 15. And you know what it looks like? It's another copy of ubiquitin, basically. A couple little differences, but this one's only going to come out when a cell is actually under attack. This is going to be one of the things we make in response to interferon, which is going to make a cell resistant to infection. You're going to have ISG15 lying around and attach that to something and you will get big responses. So all of this can be kind of thought of under this umbrella of ubiquitination, I would say. Okay, immunoproteasome, yeah, is the version of the proteasome where you change out regular beta 1, 2, and 5 for the immunoproteasome, little I version of beta 1, 2, and 5. No big deal, right? Just gives you more ways to cut a particular protein into different pieces. Because a proteasome does not just shred a protein at random. It can only cut before or after certain amino acids. So like if there's an arginine and then a serine, then yeah, your proteasome can cut it. But if you have an arginine and then something else, I don't know, a valine or something, then your proteasome may not be able to cut it there. But the immunoproteasome may be able to do that. And so it's, yeah, it just gives you a different set of peptides that you can generate from any one protein. Okay, and CTECs are cortical thymic epithelial cells. Oh my gosh. So they're a population that's going to contribute to development of T cells. They're like T cell nannies, if you like. And they're going to be found in the cortex region. Okay, so you remember beta 5, beta 1, 2, and 5. You remember beta 1, 2, and 5i. Well, get used to beta 5T. So beta 5T is another alternate version of the proteasome. And so you have, you substitute 5T for 5I. And so you're making an immunoproteasome, but with a 5T instead of the 5I there. And we call this a thymoproteasome. And so, yeah, this is going to be involved in generating peptides that are encountered by little T cells during development. Yeah, adorable. It's like a little, it's like part of the apparatus of immunological school, if you like. Okay. And so, yeah, beta 5T is going to make us a thymoproteasome, but it's still going to have beta 1I and beta 2I and beta 5T, and then all the normal proteasome stuff. All right. PA28 is the proteasome activator 28 kilodalton complex. And it is going to be induced by interferon gamma. So this is something that will be induced when immune cells are communicating with each other, different white blood cells. And it is going to replace that 19S cap. And remember, the body of the proteasome was the 20S. And so it's a substitute for the 19S cap. And so it is going to, yeah, just work faster and better. You're going to get more peptides exiting faster. It gives you a little bit quicker response. It is, yeah, just a modification that cells can make when we're making interferon gamma. When your immune system is making interferon gamma, this is a signal that the body is under some kind of pretty strong attack and something needs to be done. And so this is pulling out all the big guns. Your thymoproteasome, your immunoproteasome, and your PA28 proteasome activator complex. All right. So, yeah. Some of the things that would be tagged by ubiquitin and just degraded are going to be, yeah, defective ribosomal products, like things that you translated inside of a cell but probably shouldn't have. For example, if you get a mutation in a messenger RNA, this happens by accident all the time in your cells. You're going to make a protein, and maybe that protein has a different amino acid or two in it, or maybe it stops before it should. This is not going to be good. A protein that is misfolded will actually stick to almost any other protein, and the way your cells are regulated is by proteins that stick very specifically to each other and not to other stuff. And so if you just throw some sticky stuff in there, it's like throwing, you know, a pack of gum in with your laundry and then running it through the dryer. Yeah, it's not going to be a good result. And unfolded proteins will kill a cell. So these little products, yeah, are tagged for degradation by the proteasome, and that's fine. That's just part of the routine maintenance of a cell. Okay, so we'll cover section 6.2, and then I think we may pause after this section as well. So, all right, What's a proteasome look like? It kind of looks like a little barrel. So there's our 20S core, and your alpha subunits are right up here. Alpha 1, 2, 3, 4, 5, 6, 7. I think it's a heptamer. And beta 1, 2, 3, 4, 5, 6, 7. Great. And then you got another copy of everything on this side. And then you got 2 of these 19S caps. Now, when we are under attack and we are expressing interferon gamma, then interferon gamma is going to make us not make the 19S cap. We are instead going to make that 28, PA28, which is just another version of the cap, but it's a little bigger. You'll see. You'll see it in a couple slides. Okay, so if you've got a protein, and it is polyubiquitinated onto some protein, so a little chain of ubiquitins, the 19S cap is actually going to grab those right out of the cytoplasm, and it's going to pound them right down through this little gate, which will open up, and into the core, and then we're going to spit out on the other side peptide fragments. And I believe we can pound things in from either direction, but generally it's going to take things in one side and push them out the other side, just like a little digestive system. And these are polyubiquitins that are lysine 48 linked. There are others. You'll probably remember from the MAVS signaling, we had lysine 63 attaching to the card domain of the mitochondrial antiviral signaling protein. Does that sound familiar? The K63? That's a different way that you can hook a ubiquitin onto something. And I think there are a couple other ways that you can hook ubiquitins up that will have different meanings. And also there are sometimes different meanings if it's one ubiquitin versus a short chain versus a long chain of ubiquitins. So despite just being like little bricks that you can, little Lego bricks you can stick onto something, there's a lot of subtlety in the ubiquitin system, and generally it's very useful. I'm glad it's there. Okay, as opposed to, here is the PA28, and so look at that mighty cap up there. Wow. And look at this. So this is when it's closed, this is when it's open. It can just sort of telescope itself open and shut. And the catalytic chamber is here, so this is your normal 20S beta and alpha subunits right here. And so they're going to shred whatever protein has gone in here, and they're going to then feed it out one of the doors. It's going to open back up. Okay, and that's a good place. Let's pause this right here, and then we'll do a couple sections in the next little video.